So you can see my first slide, yes?
Yes.
Okay. Good afternoon to all participants of the conference. Thanks for the opportunity to present our experimental results about kidney cell protection during warm ischemia in partial nephrectomy. The material I'm going to present was received by the research group from Turgenev's Oral State University and also St. Luka Clinical Hospital in St. Petersburg. And as I say, Ruslan Guseinov have no possibility to take part in a conference today, so I will make it for him. And firstly, I should say that now I'm also taking part in another conference on a Baikal lake. So it is a great place, but there is one problem: not stable internet connection. So with your permission, I will show my video and so I can start the presentation. According to statistic data, kidney cancer is the 14th most common cancer worldwide. The most incidents are registered in Asia, Europe, and Northern America. Despite the tendency to growth of incidence, kidney cancer is characterized by the decrease of mortality. The main reason is an increase of treatment quality. 9 of 10 kidney cancers are renal cell cancers, renal cell carcinomas. Its evolution includes localized, then locally advanced and advanced stages. So surgery is the best treatment strategy during localized stages. It is stages T1 and 2, maybe 3. Partial nephrectomy is organ preserving procedure used in the case of relatively small tumors. The result of partial nephrectomy is determined by the number of factors, including renal parenchyma quality, quantity of preserved parenchyma, and the recovery from ischemic insult. The last is related to the preference for circulatory arrest in order to decrease of bleeding and increase of contrast between healthy tissue and the tumor. Warm ischemia is a wide using technique for this purpose. As you can see on this slide, warm ischemia leads to the negative consequences, for example, to decrease of survival rate of glomerular filtration, and, for example, of glomerular filtration rate. That's why the most preferred time of warm ischemia is no longer than 25-30 minutes. But it is obviously that the more time surgeon has, the best result of the operation can be achieved. So studies of anti-hypoxic drugs which allow to increase kidney cell viability is very actual. My colleagues from Luka Clinical Hospital in St. Petersburg have patented the use of sodium fumarate. There is a Russian patent of sodium fumarate containing drug conformin for infusion before and after operation to increase ischemic time without fatal complications, both immediately after operation and during rehabilitation. I'm sorry. So you can see that the first step is 3 droppers in the day of surgery. Of course, the surgery, and then 2 droppers per day during 3 days after surgery. Histology results show significantly less change of kidney tissue in the case of 30 minutes of warm ischemia and subsequent reperfusion with conformin compared to the experiment without any organ preservation. But some aspects of conformin for example, time before operation, drug quantity, and possible ischemia duration need to be studied and corrected. In our opinion, understanding of the mechanism of fumarate action can help to do this. In our experiments, for modeling of hypoxia, we used kidney epithelial cells line MDCK, sodium dithionite leading to a very fast decrease of oxygen concentration to 1–2% of saturated level was used for hypoxic conditions creation. After required hypoxia time, cell viability, mitochondrial membrane potential, and mitophagy level, and the rate of reactive oxygen species production were estimated. So, using ratiometric fluorescent probe MACFURA2, we determined the period of cell ability to support calcium concentration across the membrane after block of glycolysis and oxygen deprivation, so-called time to collapse. And as you see, sodium fumarate in the concentration of 5 millimoles per liter leads to an about 30-minute increase of cell viability compared to the experiment with the hypoxia without any cell preservation and cell protection. Decrease of apoptosis determined by change in morphology of cell nuclei also show protective role of sodium fumarate that is containing in drug conformin. The first possible mechanism of such effect can be related to the maintenance of ATP production by oxidative phosphorylation due to conversion of fumarate to succinate that provide mitochondrial complex 1 action and ATP synthesis by the ATP synthase. It means that in the case of oxygen deprivation, the work of complex 4 stop, and that's why ATP can be synthesized. But when we use fumarate, it can help for complex 2 work in reverse mode, and that makes possible for working of mitochondrial complex 1. And so we used NADH and FAD autofluorescence measurements to study mitochondrial complexes 1 and dithionite leads to increase of NADH and decrease of FAD signals, but subsequent addition of fumarate results in partial return to initial level. You see that there is a decrease of our autofluorescence of NADH and increase of autofluorescence of FAD. This shows that succinate dehydrogenase reduces fumarate to succinate with parallel oxidation of NADH. So you can see there is a scheme. As I said, fumarate is reduced to succinate, and so the complex 1 can oxidize NADH. But the next slide show that I'm sorry. The next slide show that this cannot provide the level of mitochondrial membrane potential level enough for ATP synthesis. Intensity of fluorescence of mitochondrial cationic probe TERMC was drastically decreased in the case of hypoxia as well as hypoxia with fumarate. In the graph below, you can see TERMC fluorescence intensity fall under ATP inhibitor oligomycin A. This shows that in hypoxia, without and with fumarate, ATP synthase is working in reverse mode for mitochondrial membrane potential generation by using of ATP hydrolysis energy. So the first hypothesis can't explain conformin protective effect. As you see, there is no ATP synthesis. There is ATP hydrolysis by complex 5 to generate of mitochondrial membrane potential. At the same time, experiments of determination of reactive oxygen species production and colocalization of mitochondria and lysosomes show same changes under fumarate. Under fumarate action. As you can see, elevated hypoxia-based reactive oxygen species production rate was reduced under fumarate action. And as you also see, mitophagy level, vice versa, was highly increased closely to control. This allows us to suppose another mechanism of fumarate action, which is related to its possibility to inhibit HIF-prolyl hydroxylases and stabilization of HIF-1 alpha in normoxia condition. This is known as one of the reasons of specialty cancer cells chemotherapy. In this case, this can activate transcription of some protective genes and metabolic pathways, including regulation of ROS production and mitochondria quality. So, high concentrations of fumarate can inhibit oxidation of HIF-1 alpha, and so and can start some mechanism of cell protection in hypoxia during warm ischemia. To check this hypothesis, we have determined reactive oxygen production rate and mitophagy level in the presence of oxygen and addition of fumarate. So we did not use dicyanide in these experiments, and we wanted to know how these parameters can change under elevated fumarate concentration. As you can see, both parameters change in the presence of fumarate. That allows us to speculate about the ability of HIF-1 alpha stabilization. You can see that under fumarate action, we can see the decrease of production, and also we can see the increase in the level of localization of mitochondria with lysosomes, and it may be as a possible signs of activation natural mechanism of cell protection during ischemia. But of course, this needs some more experiments, for example, Western blot analysis, and maybe the rate of HIF1 alpha gene expression, and so on. But now we can see we can say that the second mechanism of protection may be the most preferable explanation of fumarate protection during warm ischemia. On slide, you can see the main conclusions of the presented study, which I have already told. Thanks you for attention, and I'm ready to answer your questions.